Greetings, I'm Yvonne, Staff for Science for the Public, and I welcome you to Contemporary Science Issues and Innovations. Today, we'll get background on how viruses infect us and how vaccines are created. In the COVID-19 pandemic, mRNA vaccines have been introduced for the first time. We'll hear about the potential merits of these vaccines as well as the traditional DNA type. Our guest is Dr. Florian Douam, Assistant Professor of Microbiology and Investigator at the National Emerging Infectious Diseases Laboratories at Boston University School of Medicine. Excuse me. Dr. Douam specializes in human immune responses to a range of viruses to collect information needed for vaccine development. It's a great pleasure to welcome Dr. Florian Douam. Thank you very much for joining us.
Thank you for having me.
Okay, and I'd like to start by just getting a little bit of general background. As you probably know, viruses and bacteria are very commonly confused. And so people run to the doctor asking for an antibiotic for a flu infection or something. Could you please just give us a wee bit of background on that, the difference, please?
Yeah, of course. Bacteria are living cells. You could see them as unicellular organism. They can multiply, replicate by themselves outside the human cells or outside the body. So they are really, you can really see them as a single, unique, living entity. And they don't need the human cells or human body to. Viruses are really different. You can see them as an assembly of protein that's encapsulating, protecting a piece of genetic material. The major differences with bacteria is that you can see them as obligate intracellular parasites, meaning that they really need to get into a cell to be able to replicate and propagate. And they can infect human cells, and they can actually also infect bacteria. And so the reason we say obligate intracellular parasite is because they're going to subvert, kind of hijack the cellular machinery to be able to replicate.
Right, I see. And although these viruses are not really living things considered life forms, they are very, very clever. And you're going to be able to explain that to us a wee bit. First, could you take us through the steps about identifying a virus like COVID-19 is the big one right now, but you look at a lot of them, just identifying that virus and then how it infects cells for us?
So let me jump back to what you just said. So actually the question on whether viruses are a form of life or not is very much debatable. I am one of the virologists that consider that actually viruses are a form of life.
Aha.
So yeah, this question is very much debatable because it depends on what definition you give to life, right? What's defining life? And over the past 5 to 10 years and even a few decades, we have found that viruses can also infect viruses. So we have viruses that can infect viruses, and people have been using this as a support for, saying that viruses may be different life form, but still a life form. Another argument toward their living ability is the fact that they can evolve over time, right? Virus mutates and evolve. They are subjected to natural selection, like bacteria or other animal species are. But if I want to be fair, the reason some biologists also say that viruses are not alive is directly dependent on the fact that they cannot replicate by themselves, right? They need a human cells or bacterial cells to replicate. So it's very much debatable. I would say myself that they are alive, but I completely acknowledge counter arguments as well. To come back to your question, so you were saying, can you remind me? So
Yes. First of all, thank you for that clarification. I didn't realize that scientists are now re-examining this thing, that viruses, they should be reevaluated in regard to their biological status here. Thank you very much for that. And then the next question was, could you help us go through the steps of like identifying a virus, which is a big thing, like in the COVID-19, as it's commonly called now, and was part of the SARS group and so on, but just identifying how they identify a virus and then determining how it infects cells, which I think is one of your big things.
Yeah. So, I mean, one of the first steps you do to identify a virus is you identify patients that have an unknown disease or suspected viral disease. You get samples from these patients, and then you try to sequence viral genomes. So, meaning we try to read the genetic material of the potential pathogens inside these patients. And then once you get this genetic material and you have the sequence of this genetic material, you can compare it with all the genetic material available from other viruses. And this is what happened for SARS-CoV-2. They sequenced it and they compared it, and they found that it was closely related to bat coronavirus and to SARS-CoV-1. And then so that's kind of the first step. But in parallel, you also take these patient samples and you put these patient samples on cells. So you have, like, you see this as a plastic dish in the lab that contains cells. So it's a monolayer of cells, and you just drop these patient samples on these cells, and you see whether the cells die, or you see whether the amount of genetic material of the virus increase over time in this dish. And then you confirm again by sequencing that this is still the same genetic material that you found in patients, which allow you to first say, yes, this is the same virus, and yes, it's replicating in human cells.
One of the confusing things that probably has come up with this virus, this recent virus, is that the question of DNA and RNA viruses. Could you explain the difference between those for us?
Yeah. So viruses can be classified into different, categories. And the two big categories that we use to classify viruses are DNA and RNA viruses. So DNA viruses, they include very well-known famous viruses like hepatitis B virus, smallpox virus, herpes virus. And the main differences with RNA virus is that, when these viruses infect the cells, the DNA template, the DNA genome that they carry, first needs to be used by the cells to make RNA, right? So this DNA makes RNA, and this RNA then makes proteins. And so these DNA genome are usually larger than RNA virus genome, and they are also less susceptible to mutation and change over time. Striking characteristic of DNA viruses too is that they are more prone to latency stages. We induce latencies in human cells, meaning that you have this DNA persisting into the whole cells. And that's also one of the reasons, that's not the main reason, that's one of the reasons why you have a lot of these viruses that can cause chronic infection, right? For example, if you take HBV, you have this DNA template from HBV that can persist over time in human cells, and it's very difficult to it's impossible for the organism to kind of get rid of this DNA, so it's going to persist. And that's the same for herpes viruses, right? You can live with herpes viruses all your life. So you could see these DNA viruses are big viruses that often are associated with chronic infection, where the DNA persists into the small cells, and where you first need to make RNA from the DNA to synthesize viral proteins. So RNA viruses, on the contrary, they have usually smaller genomes. And when the RNA virus infects the cells, then the RNA is directly detected to the cells, and you have this RNA directly making proteins, right? So you don't have this DNA making RNA and then this RNA making proteins. You already have RNA making proteins immediately. And so because a striking characteristic of RNA viruses too is that they are more susceptible to mutations. The viral proteins that replicate the viral RNA makes a lot of mistakes. And that triggers a lot of mutation over time. Although DNA viruses, as I said, they are less susceptible to mutation because they have this kind of DNA is more stable, and they have more checkpoint when making mistakes. And also another characteristic of RNA viruses is that they are mainly associated with acute brief infection, unlike DNA virus that I was just mentioning, more chronic infection, because these RNA molecules are also more fragile, and so they do not really persist into the host cells. A very unique, very interesting exception is HIV. HIV is an RNA virus, but it's an RNA virus that can make DNA from the RNA.
Whoa, can you go through that again? I didn't realize it can make DNA from the RNA.
Yeah, exactly. So that's what we call retrovirus, meaning a virus that has RNA, and the virus uses its RNA to make DNA, and then incorporate its DNA to the genome of the host cells. And that's why HIV is persistent. It's persistent. So HIV is some kind of example where you actually have an RNA virus that is a chronic virus and it's chronic infection because it can make DNA from its RNA. So yeah, I just wanted to let you know about this special case because there's always exception to the rules.
That's really a challenge, then, isn't it? It seems like that's a challenge to deal with that. I don't know what the identification, but especially would that have an effect on mutation rate or the viability of these mutations, the success of those mutations as well?
Yeah, that's why it's so difficult to fight HIV, because you have a RNA genome that is highly prone to mutation, and then transfer all of this mutation into a DNA segment that integrates into the host cells. And then because of this, the virus can find some kind of hideout places into the body to hide from antiretroviral therapies. So that's why these antiretroviral therapies, they kind of blunt viral replication, but if you stop treating the patients, the virus comes back. Because all of this hiding DNA kind of gets reactivated. So that's the major challenge in HIV field. And because it mutates so much, you almost have one HIV virus different per individual. That's also why it's so difficult to make vaccine against this virus.
I see. Now, this is not all viruses. This is some viruses that work in that way. How do these viruses actually infect the cells? We've seen these spike things with the coronavirus and so on, but viruses they're almost beautiful. The structure of these things are beautiful, but their ability, the way that they penetrate the cells differs, correct? Is that correct?
Yeah.
So the coronavirus has this spike thing, but the other ones have other mechanisms.
So each virus has its own what we call envelope proteins or surface proteins, because you are going to have viruses that are going to have a shell of lipids, and this shell of lipid is going to have this envelope glycoproteins, envelope proteins. And you also have viruses that do not have envelope of lipids, that directly have cell surface, that directly have like viral proteins at the top of their shell, right? So you really have two types. You have either a shell of proteins with a viral protein on top of the shell of proteins, or you have a shell of protein encapsulated with a lipid envelope into which there is viral proteins. And so all of these proteins that are exposed at the surface of the virus, whether they are exposed into the shell of protein or this lipid membrane, they are going to infect cells by recognizing specific cell surface host protein. And this is going to be a very specific interaction between a cell protein and these viral proteins. And this specific interaction kind of determine where the virus can get into which tissues, and where it cannot. For example, if I take the example of hepatitis C virus, we know that the envelope viral protein of hepatitis C virus only recognize a combination of cell surface proteins that are present in the liver cells, right?
Yes.
And so for the coronavirus, SARS-CoV-2, we know that this SARS-CoV-2 spike protein recognizes a receptor called ACE2, and this receptor ACE2 is not present everywhere in all tissues of your body. We know that it's mainly present in your nasal cavities and in your lower respiratory tract, which kind of define in some way the cell tropism where the virus is going to go and infect cells. We kind of think, there's some evidence that ACE2 is also expressed at different, at other locations of the body, but it's still unclear, and we are trying to understand that better. But you see this as this first interaction between the viral envelope proteins and a cell protein at the surface of the cells that is really going to initiate the infectious cycle of the virus and guide where the virus is going to infect the cells and replicate, or where it's not going to be.
I see. Then you do a lot of the work that leads up to looking for a vaccine or developing a vaccine. You lay all the groundwork, I guess, for that. But in this present pandemic, we've been introduced to mRNA vaccines, and they're more typically DNA vaccines. And I recall an article in which you discuss these differences. Could you give us some background on that? What is the difference? There seem to be some questions about the viability or preference for these two.
Yeah, so let me first kind of define what you can call traditional vaccine. So traditional vaccine, I would put them in 4 categories. First is soluble viral proteins. So you would just use the protein of the virus, make it soluble, and inject and vaccinate a human with these soluble proteins. A very common type of vaccine is using a virus shell, right? You would just use this shell of the virus, and inside these shells, you're going to put either virus protein or you're going to put a piece of a viral genome. And then this virus shell is going to take into a cells this virus protein or this piece of viral genome. The cells will express this virus protein or this piece of virus genome, and then the immune system is going to recognize that. Another type of traditional vaccine is what we call weakened virus. So it's literally you take an infectious virus, a virulent virus, and you passage this virus into human cells for a very long time to create a weakened virus. And then this virus is going to be able to trigger an immune response, but it's going to be too weak to develop disease, create disease. A very good example of this are the yellow fever vaccine, the measles vaccine, and
Smallpox?
Sorry?
What, smallpox, one of those?
Vaccinia virus, yeah. Vaccinia vaccine, the vaccine of smallpox, yeah, exactly. Right. And the last type of traditional vaccine is what you would call a dead virus. So it's a virus that you kill. And so the shell of the virus is going to be recognized by the human immune system. So that would be the 4 types of traditional vaccine that we have. In the context of SARS-CoV-2, I listed you this virus shell to transport a virus genome. So that would be the AstraZeneca vaccine or the Johnson & Johnson vaccine. That's typically what they do. They use a virus shell, so from an adenovirus, They emptied this virus shell from the adenovirus material and they replaced it with a piece of SARS-CoV-2 genome. So now coming back to the mRNA vaccine, the first thing I really want to say is that this vaccine, this vaccine technology has not emerged over the past few months. This technology is the result of almost 50 years of fundamental basic science. And it's very important for people to understand that, because I've heard often that, this mRNA vaccine it's a miracle, and it's actually something that we have been working on for 50 years. And I think one of the first discoveries that I can remember of that led to this mRNA vaccine at dates of 1970s. So it's really a long path. And I would say that fortunately, when the pandemic started, we had this technology mature at the right time. It was ready. And so in terms of more like fundamental aspect of what it is, so see the mRNA vaccine as a lipid shell. So it's not a virus shell, it's not a protein shell, it's a shell of lipid that will transport a piece of virus genome that is encoded into RNA. So you're going to have this lipid shell that is going to shuttle a piece of virus genome into a cell. And in the context of mRNA vaccine for SARS-CoV-2, it's actually a piece of RNA that codes spike proteins, right? So there's no infectious virus. It's safe because it's only spike that is produced. It's not other pieces of the virus. So you have these lipid shells that go into the cells and ask the cells to use this RNA to produce a lot of spike, and spike is then secreted out of the cells. And then your immune response is going to recognize spike. So that's the main, I would say the basic principle. The huge advantage of this technology is that you can use this technology to express anything in the cells. A huge advantage of this mRNA vaccine in comparison to, for example, the Johnson & Johnson AstraZeneca vaccine that uses virus shell is that because it's a protein shell that these vaccines are using, right? I'm talking about the AstraZeneca and J&J. It's a virus shell. The immune system is also going to recognize this virus shell, right? So the immune system is going to mount an antibody response against this virus shell as well as spike. So that means that if we were to, confront another coronavirus pandemic in 2 years, we would not be able to use the same virus shell because the immune system would immediately recognize the virus shell. And that's why AstraZeneca and Johnson & Johnson have been using virus shell that are for which humans have a very low pre-existing immunity against, right? For example, AstraZeneca has been using an adenovirus shell from chimpanzee, right? Because humans they don't have pre-existing immunity against. The huge advantage of mRNA vaccine is because they use this lipid shells are not recognized by your immune system. So if we were to have another coronavirus pandemic in a year or two, we could still use the same lipid shell.
I see. So roughly the mRNAs are very adaptable. You can adapt them rather quickly, but there are some maybe disadvantages in that they are, I'm not clear on this, are they unstable in certain temperatures? That's the one of the big issues, I think.
Yeah.
I don't know if that's true.
Yeah, so that's a good point.
Yeah, okay. So, and maybe require 2 doses instead of one. I'm not clear on that either, but it's just the good thing is that they're you can adapt them quickly. You can get them going quickly. I see.
Yeah. So coming back to this low shelf temperature, yes, that's a limitation. But, mRNA vaccine, it's really 50-50. Like 50% of the success of the vaccine is going to be the formulation, so the lipid shell, and 50% is going to be the mRNA. And they're going to be, I believe, in the future, huge improvement to improve the lipid shell formulation to increase the stability of and the preservation of this mRNA vaccine. And we're already seeing this because we know that the Pfizer vaccine requires lower temperature than the Moderna vaccine. So this is just the beginning of the technology, and we could expect that, we're going to be able to increase the stability of this technology over time. Regarding the second shot, this is, I would say, the major challenge, right? To me, that's really making an mRNA vaccine that requires a single shot is a major challenge.
Mm-hmm.
But I would say that people were probably not expecting before the pandemic the 2 doses of mRNA vaccine to be that protective. Because what I said to an interview at BU a few months ago, I was talking about mRNA vaccine being kind of the first guard, the first troops to kind of, confer some initial protection in the population before this more protective, more immunogenic vaccine coming, like the adenovirus, G&G and AstraZeneca. But what it seems is that actually, and I can revise kind of my discussion on this, because these mRNA vaccines actually are quite potent, and the adenovirus vaccine actually might still also require 2 shots.
Mm-hmm.
The same. It seems that they need 2 shots. That we need 2 shots from these vaccines to reach the same kind of efficacy. So yeah, I think mRNA vaccines are actually, knowing more about it, I think they are actually even more promising than what I thought.
That's very interesting. Maybe the best thing is that it's like a rapid response, and that I'm eager to get to some of your work. And I would like to ask first, though, a couple of things that, first of all, this can be kind of deployed quickly, the mRNAs. And by the way, thank you for bringing up that this has been kind of on the books for a very long time. There's a very interesting story about the scientists who were trying to develop that over decades. And one of them actually got demoted because she couldn't get it going. So I'm happy to see this, but it's a kind of typical story in science. But another thing here is that this was put on the market very fast compared to the usual phase trials of vaccines. Do you anticipate that we can bypass those lengthy phase trials in the future, or is that just applicable to DNA viruses, vaccines rather?
So usually it takes 8 to 10 years from development to commercialization and licensing to vaccine. So I completely understand why people, why some people have been very skeptical, and wow, this has been very fast, right?
Yes.
And we have been waiting for 50 years for HIV, 40
That's exactly
years
right.
for HIV vaccine, and there's no vaccine against hepatitis C virus.
Yes.
What's happening, right? So the big things that we need to understand here is that the limitation is often phase 3, because phase 3 clinical trial requires tests on tens of thousands of people.
Yes, I see.
And what happened here is that it's a combination of 2 things. First, this mRNA vaccine, right? We can make them very quickly, right? In 60 days, in the context of SARS-CoV-2, we had the mRNA vaccine ready. But the major thing here is that for phase 3 clinical trials, we had access to as much patients as we wanted.
Yes.
And that's exactly why it was faster. It was faster because people cut corners. It was faster because we had all the patients that we wanted to test the efficacy of this vaccine faster. And this is why, vaccine like, for example, Zika, when Zika came out, it is why there is still not a licensed vaccine against Zika, because it's very hard to find 50,000 people affected by Zika. And this was the, and I can cite many other examples, and for Ebola, we are just getting on for vaccine, but, it took also so much time because, 50,000 people affected by Ebola would be dramatic.
Yes.
Not something we see. HIV and HCV are a different of different concern, right? They have been around for decades. Millions of people are infected by this virus.
Yes.
But the challenges of developing vaccine against this virus is that we cannot find infected patients is the fact that this virus mutates extremely frequently. Their mutation rate is extremely high, much more than the coronavirus, right? So that's a different challenge.
Faster than the coronavirus.
What?
Faster. That
Oh,
the,
yeah.
oh, so the mutation is much faster in HIV.
Well, it's. I would probably refine this. I would probably not say faster. I would say that the inter and intra variability, the inter and intra individual variability.
I see.
Higher, right?
I see.
So we have much more different types, different variants, if you want me to use that word, much more different variants around, different type of viruses. The genetic divergence between the different type of HIV and HCV is extremely high, meaning that if you were to make a vaccine against one variant, you would probably protect person A, but not person B.
I see.
And
I see.
although for SARS coronavirus, yes, we have different variants, but we can still generate vaccine that would be, that would cover most of the variant.
Yes, I see. That's very good information. Thanks. And then another very quick one, and then I want to go to your work specifically, is that we hear a lot at the beginning of this, we heard, and we still hear herd immunity. Don't worry about a thing. We'll have herd immunity as opposed to vaccine immunity. And I understand that you need a very high rate of vaccination in order to get vaccine immunity. So those two things, the difference between them, please, and what's the percentage of population you need for the vaccine immunity?
So herd immunity is a very general term. And if you look at for history and the influence of vaccination, actually most of the herd immunity built over history were actually caused by vaccine immunity.
Oh.
So vaccine immunity makes herd immunity most of the time, actually. So these are very connected concepts. So herd immunity is when a sufficient fraction, of a population is protected against a disease, right? And
yes,
it can be built by vaccination. And people who are exposed to virus, and are not protected. And so if you want, that's literally one building the other one. Vaccine immunity is when people are immune to virus because of vaccination, and this participates in herd immunity.
Yes. Right. And that you need large numbers in order to achieve that. But that I wondered about, we were getting almost this mythological thing about herd immunity. Don't worry about a thing, don't need masks and so on. But it certainly never worked for things like smallpox outbreaks. There was no such thing as herd immunity. We go with vaccinations, which is exactly your point, I guess. Then thank you for that information. And now I would like to go to some of your work. And you cover, or you do work on a number of viruses, I believe, and you use humanized mice. And I'd like very much to know the advantages of that and how that works.
Yeah, so humanized mice is a concept that we use to define mice that would carry human genes, into which we will incorporate human genes or human tissues, or both. So the fundamental idea beyond the humanized mice is that in virology, we are often confronted to three issues when you want to understand how the human responds to a virus infection. The first issue is that many of the viruses that are extremely highly clinically relevant for human health only affect humans. Right? For example, and only affect humans in specific species of primates, I would say,
Yes.
to be complete. And for example, I take again the example of hepatitis C virus or HIV, right? They only infect humans and chimpanzee and probably other species of higher primates.
Yes.
And so if you want to study these viruses in a laboratory and you really want to understand how the human respond to infection, right, you need to use animal model. And, there are extremely high ethical challenges now associated with the use of higher primate in research. And this is also associated with very high cost. So what you need a small animal model, scalable animal model, into which you can recapitulate human immune responses to this infection and a human in a mouse model that can, replicate these viruses. So this is what the humanized mice, serve for. You can engraft these humanized mice with human tissues, and then you have mice that becomes permissive to HIV or HCV. So that's the first point. The second point is that there are some challenges associated with using human patients, right? Because you could say, you identify a cohort of human patients affected by HIV, right? And you get samples from these patients, the blood samples, human immune responses. But the issue with these patients is that you only get blood, right? Only get access to the blood. And we know that many, many important events during a virus infection actually happen in tissues.
Right.
So, and you cannot ask HIV patients to give his lymph nodes or things like this, right?
Yes.
So they are also, because there are a lot of ethical considerations. So you see, these two major important points clearly highlight the fact that humanized mice are right now kind of the only tool that we have to study viruses that only affect humans and study the human immune responses to these viruses into a cost-effective small animal model.
Yes.
And so I was telling you the example of HIV. Humanized mice have been absolutely instrumental for understanding HIV, developing antiretroviral therapies. The way we are doing it is we can engraft mice with a human immune system. So cells, so human cells that are components of the human immune system. And because we know that HIV targets cells of the human immune system, we can actually render these mice permissive to HIV, injecting into mice these human cells. For hepatitis C virus, we can actually replace a segment of the mouse liver with a segment of a human liver. And then we can make the mouse liver becoming infected, actually the human cells inside the mouse liver that becomes affected. So these mouse models are absolutely instrumental. And we are using them in the lab very routinely to understand, human responses to viruses in human tissues.
Yes, that just sounds like it would really accelerate two things from the naive point of view here, that first of all, it would give you faster insight into how the mechanisms of a virus, first of all, and then secondly, a more effective pathway toward developing a vaccine because you have a better understanding of it. Is that true? And then is it fairly easy to do these kinds of, I guess you'd call it humanization of the mice? Is that relatively easy?
Well, it depends what you mean by easy. So interestingly, in the humanized mice, humanized mice have been under development for the past 20 years. And we are all aware that we are never going to be able to develop the perfect model, right? Mimicking the perfect human immune responses. So these are absolutely instrumental tools, but we also need to be aware of the limitation, right?
Yes.
But in terms of generation, it depends what you are actually trying to make. If humanized mice that are engrafted with a human immune system are fairly easy to make. But what it's harder is actually making sure that the human immune response in these mice mimic or resemble what you see in human.
Yes.
So the challenge is not the engraftment. The challenge is making sure that your human immune system is going to behave the same way than a human patient.
Yeah.
So that's something we are working very hard on, us and other labs. And we have made significant improvement, I would say, over the past 15 years. In one of our recent work, we have, for example, shown that in one of a novel humanized mice model we developed, we are able to recapitulate human immune responses that are similar to human patients' immune responses when they are vaccinated with the yellow fever vaccine.
Right.
So we have found like clearly similar immunological signature between our humanized mice and human patients vaccinated with the yellow fever vaccine. So that's promising, and that's telling you that yes, we are getting closer to a stage where we can really use these mice to better understand human immune responses to vaccine,
Yes.
into infection. So in general, to kind of make the switch to SARS-CoV-2, and that's really our current focus right now. We have been using mice that are co-engrafted with the human immune system and human lung tissues.
Yeah.
SARS-CoV-2 has been challenging because, as you probably know, you cannot infect conventional mice, laboratory mice. Laboratory mice are not permissive to infection.
Yes.
And this is to come back to what we discussed earlier. This is due to the fact that the mouse S2, so the receptor of the spike, the mouse S2 doesn't recognize very well the spike of the SARS-CoV-2. So this has been challenging. So what we did is that we generated mice that carry a human immune system and also human lung tissues. And what we found is that if you inoculate these mice with SARS-CoV-2, these mice actually develop some severe features of some feature of severe COVID-19 disease in their lungs.
Yes.
So right now we are using these humanized mice in the context of SARS-CoV-2 to understand what makes the human immune system in some time being able to clear the virus while protecting the tissue, and in other case, clearing the virus while damaging the tissues. And so we are trying to identify the differential immune responses that in one case damage the tissues and in other case protect the tissues. Because we know more and more that the reason patients develop severe COVID-19 disease is actually not because of extensive viral replication, but because of overreaction of the human system. So we are using our mice to understand and to capture immunological signature that define tissue protection versus tissue damage. And by doing this, we can harness the knowledge to potentially develop therapies that would protect or help the human immune system to further protect the tissues during severe COVID-19 infection.
Right. This is very big challenge, but I know that's most interesting, but you've got a lot of work there, I can see. Dr. Douam, I am sorry we are out of time, and I had hoped we could get more about you, because you investigate a number of viruses, and this your approach is of particular interest. So maybe we'll ask you to do it again.
Yep.
It's this is very interesting work at a very important time, because we're going to see more and more viruses, and what you are doing is right on the target, apparently, the way to investigate these things. We very much appreciate your expertise and your time today. Thank you so much.
Thank you very much.